Yes, my presentation today was about the use of haploidentical stem cell transplantation as a tool for immunotherapy in children with acute lymphoblastic leukemia. So our focus is mainly to use allogeneic or haploidentical stem cell transplantation as an immunotherapeutic approach, and haploidentical means we use in most of our children, we use the parents as the stem cell donor. And in order to facilitate the transplant and make it safe, we do in vitro T cell depletion or ex vivo T cell depletion. So we take out the T cells, which would otherwise cause graft versus host disease, out of the graft, and T cells which we take out are the so-called TCR alpha beta T cells, because these are the main T cells which cause graft versus host disease. So this allows us to use parents as a donor in our children, and then we, after the transplant, we further treat these children with an immune augmenting therapy against the leukemia in order to avoid relapses. So our focus is on monoclonal antibody against the target antigen CD19, which is expressed on all the leukemic blasts, or on most of them. We have generated our own chimeric anti-CD19 antibody, which we use then for these children after they have received these stem cell grafts from their parents. So far, I think our results are promising. So we have done a number of children now who have relapsed after a standard transplant. Then we do a second, or some children even received a third transplant from their parents. And then we do this antibody therapy against CD19 for up to 2 years. And so far, our overall survival is quite promising in these children, in contrast to our previous experience without this antibody treatment after transplantation. And the main focus is to activate the so-called natural killer cells with this antibody. So this antibody is engineered to exert a very effective so-called ADCC, antibody-dependent cellular cytotoxicity, which activates quite effectively natural killer cells via their Fc receptor, the so-called CD16. And this activation results in quite long relapse-free survival of these children. And the tolerability is very good. The children tolerate this treatment very well. We have not seen severe side effects of this antibody treatment, and are now, of course, focusing on performing a multi-center prospective trial using this approach in patients with advanced leukemia. So our other focus is on the use of so-called memory natural killer cells. This is an interesting subpopulation of natural killer cells which seems to have some memory, like CD8 memory T cells. There seems to be a similar feature in NK cells. So once the NK cells have seen the leukemic cells and they see it again, they seem to have a memory for them, because they are much faster activated, they are much more effective, and they produce much more cytokines after second exposure to these leukemic blasts. So we are also focusing on using this kind of memory NK cells for post-transplant adoptive transfer, so to isolate these memory NK cells and later adoptively transfer these NK cells, so these are donor NK cells after transplant, to adoptively transfer these NK cells to the patient, also to avoid or prevent relapses in these high-risk patients. That's the one approach, and the other approach is that we use also a CAR T-cell approach. Our approach is a little bit different than from the other approaches, so we use an adapter strategy, so we separate the CAR T cells from the antigen, that we use a CAR T cells which is directed against a biotin. Once the biotin is bound to a protein, in this case an antibody, so we generate one CAR T cell against the biotin, and then we can use any biotinylated antibody as the target. Of course, this is a lot of still ongoing work in vitro and in vivo in mouse models. It seems to work quite effectively, but we have not yet clinical data. That's hopefully this or next year we hopefully can start with some clinical trials. But the system is very flexible because we can use several targets at the same time. The problem at the moment with CAR T-cell therapy is that, especially with CD19 CAR T-cells, which is mainly used in acute lymphoblastic leukemia, is the fact that relapses occur, and then these relapses are CD19 negative. So there is a selection process. We can use different or several targets at the same time. For example, we use biotinylated antibodies against CD19, against CD20, against CD22, which is all expressed on the leukemic blast. And once we use all this together, it seems that at least in our in vitro models, this antigen escape is much less of a problem than we use only one single target antigen. So that's, I think, one of the advantages of using such a flexible system. And of course, we also anticipate that the system is safer because we can switch the CAR T cells on and off, because these CAR T cells otherwise do not recognize anything unless you don't give any biotinylated antibody. But we have to await, of course, further clinical studies.